This video covers the IGCSE Biology Objective 5.1.9 on the 2025 CAIE syllabus to explain the effect of changes in pH on enzyme activity. So in the last video we looked at temperature, and now we're going to look at pH. But before we get to that, let's just very briefly go through the pH scale. The pH scale is a scale that is used to measure the acidity or alkalinity, in this case labeled basic, level of a solution that goes from 1 to 14. 1 to 6 is an acidic solution, 7 is neutral, 8 through 14 is basic. So the lower the number on the pH scale, between 1 and 6, the more acidic a substance is, and the higher the number, between 8 and 14, a solution is, then it will be more basic. So that's the essence of a pH scale. And so some examples of pH are listed here. So in your stomach, we have hydrochloric acid that measures at around a pH of 2. Something like tomato juice is a pH of 4. Human urine, which is fairly neutral because it's mostly water, but then let's not forget there are some things in there that will make it slightly acidic. Pure water is a pH of 7. Human blood is almost neutral with a pH of 7.4, some slight alkalinity in there due to the protein structure of the human blood. Something like hand soap is a pH of 10, and something like bleach is a pH of 12. So this is just to have some idea of where the range of pH falls in some commonly known items that we might be aware of. All right, so when we're looking at pH on enzyme activity, we will see a graph that looks like this, where we have pH range and then the rate of reaction. And there are 3 points on this graph that we will be discussing, A, B, and C. We will discuss B first and then points A and C together, just because the same thing is happening in both of those particular points on the graph. All right, so point B, which we call optimum pH, and the pH can range from 1 to 14. It doesn't have to be 7 every time. It's just where this structure of the graph lines are placed according to the pH scale. So it's not going to always be 7. Different enzymes have different optimum pHs, but this is the peak of the graph, point B. So this is going to be where enzyme activity is the highest, and this is going to change for different types of enzymes. So we have an enzyme in our stomach that's going to be optimum of 2. Why? Because our stomach is a pH of 2. That enzyme will not be able to function in places very well that are of a different pH. And so the closer an enzyme is to its optimum pH, then the faster the rate of reaction will be. So the lower and higher pHs, other than optimum, which are depicted by the A and C on this graph, are the ones where we're going to see slower rates of reaction for that specific enzyme. So if its optimum pH is 7, then anything between 1 and 6 and 8 to 14 are going to slow it down. And the farther away you get from optimum, the slower the reaction is going to be. And so that's why the graph has this particular shape to it. So what's happening is, as the further away you get from optimum, that is at this parts A and C on the graph, the active site's shape is changing, and therefore you're going to have less and less and less activity. So the further away you get from optimum, the more the shape of the active site changes. As a result, we know that's called denaturation, and eventually the enzyme will stop working because it can no longer bind to its substrate and the chemical reaction will stop.